Welcome, everyone. Welcome to Strait Science. Strait Science is an evening seminar series put on by the University of Alaska Fairbanks Northwest Campus and also University of Alaska Fairbanks Alaska Sea Grant. And both UAF Northwest Campus and Alaska Sea Grant, we are public servants of the Bering Strait region. We serve all peoples of the Bering Strait region, which is the homeland and waters of the St. Lawrence Island Yupik, Inupiaq, and Central Yupik peoples. So tonight, we have a talk on harmful algal bloom activity in Western and Northern Alaska. This is something that we were ground zero for, so to speak, in 2022 when the region, especially up in the Strait, experienced the largest, most persistent, most toxic bloom of a harmful algae called Alexandrium. And we're grateful that we have researchers and everyone in our region is interested in learning more about this situation and how to respond and the research that's going on. So tonight, you couldn't ask for a better lineup. We're sorry right off the bat that Kathi Lefebvre, who is with the NOAA Northwest Fisheries Science Center in Seattle, and she is the director of the Warren West Lab, she has been sidelined by the current government shutdown. So she is not going to speak tonight, but I'm sure she's here thinking about it. So we will lead off with Don Anderson. Don Anderson is a world authority on harmful algae, and he's been studying harmful algal blooms for decades, just a few. And the last 7 years, he has started to pay attention to the North, and we are grateful for that. He is a senior scientist in the biology department at the Woods Hole Oceanographic Institute in Woods Hole, Massachusetts, and we're grateful you've traveled all this way. And bringing it all home tonight will be Patrick Charapata. He was recently a graduate student of Kathi Lefebvre's, and he is very successful and has already become a researcher with the Georgia Aquarium, and he will tell us more about that. With that, we'll have our questions at the end, and I'll be monitoring the chat and whatnot. There is unusually here in Nome, we have food in the back, so knock yourselves out and munch all the way through this. I don't think people will be able to hear us crunching on things. And take it away, Don Anderson. We're so glad you're here.
I think I just turned it on. All right, good. Well, thank you, Gay. Thank you very much. And I want to repeat Kathy's regrets at not being here. Kathi is, as anybody who knows her, so full of energy and enthusiasm. It's wonderful to give talks with her or to listen to her talks. And sadly, we won't have that tonight. But we do have some interesting things to tell you. This is the third time I've given a Strait Science talk. I feel a little, I suppose, embarrassed to hear that there's apparently some major storm coming in. Rick Toman can maybe tell us about it, coming in tomorrow. 3 years ago, I gave a Strait Science talk when your big storm Merbach was actually happening. I remember Gay was doing this from her house or something, and the windows were rattling, and I'm on the East Coast. And everybody was listening to a talk in the middle of a huge storm, but I guess you've got another one tomorrow. In any case, what I want to go through is some of what we've said before, but give an update on what we have learned in more recent times about the harmful algal bloom problem in this, basically the Bering Sea and the Alaskan Arctic. And to do that, let's start at the beginning. And for those who don't really know what harmful algae are, even what algae are, let's start there. This picture is, if I just took some water, concentrated a little bit from the ocean, concentrated it, put it under the microscope, what you see are all these little cells. most of them are tiny plants, literally like the blades of grass that on land, this would be tiny plants in the ocean. And some of them, like you see, these little golden balls have pigments, and those pigments then are used to harvest the sun's energy with photosynthesis and basically grow more cells like these. And that's really the base of the marine food web. It's also these cells produce half of the oxygen that we breathe. And so algae in general are hugely important to all of us. But among the thousands of species of algae in the ocean, there are 100 or 150 or so that cause harm in different ways. And some of those do that by producing toxins, very potent toxins. And in our field, we have these acronyms for the human poisoning syndromes that come when humans, or in fact other animals, eat shellfish that in turn have been feeding on those algae, concentrating their toxins in the tissues of the shellfish so that they can be very dangerous. And the different species produce different toxins that cause different symptoms. The one I'm going to talk about today is paralytic shellfish poisoning, or PSP. Another problem that occurs here in Alaska, but not as severe, that I won't talk about, is amnesic shellfish poisoning, or ASP. This one, relatively new to the field, first discovered back in around 1987. And interestingly, if you ate shellfish like this, contaminated with this ASP toxin, it can kill you. Many of these are fatal syndromes, but if you survive, you would have permanent memory damage, permanent brain damage in the part of your brain that store your short-term memories. So just a meal of shellfish that's why it's called amnesic shellfish poisoning. But we're going to focus on PSP, or paralytic shellfish poisoning. And in the rest of the world, in the far, part of this country, the East Coast, and many parts of the world, basically we worry about shellfish, clams, mussels, scallops, and so forth, accumulating this toxin and then passing it along to human consumers. There is also a concern about crabs. And that's where most of our monitoring, management, and so forth has been focused. But out here, we have this whole world of a much more diverse food web, and people that rely on that food web through subsistence. And all of these various elements of this food web are of concern. And of course, we didn't know when these projects began very much at all about the movement of these toxins through that food web. And that's what Patrick will talk about when I'm finished with sort of the oceanography and the biology of this problem. So speaking of the oceanography and the biology, this is the culprit. This is Alexandrium catenella. These are two cells here. It's a dinoflagellate, and these cells are like the smallest point of a pin, just barely visible to the naked eye. And these cells down here are also that same species, Alexandrium catenella, but they are a resting stage, or a cyst. And so if you go over on the right here, you see the life history or life cycle of this organism. And let's just start down here with these cysts that are in the sediment. And up here, the cysts are in the sediment throughout much of the year, and that's true most areas. And the blooms are only a few months of the year. The rest of the time, they're just dormant or resting in the sediments. And when conditions are right, we can talk a little bit about that, they will germinate, they'll hatch. And then one cell makes two make four, and you keep going, and you make more and more cells. And that then produces the dangerous situation with a lot of cells up in the surface of the ocean, making the toxins that are then entering that food web. But at some point, those cells then form gametes, these small cells here that fuse together and produce a zygote that comes back to the sediment and forms that cyst. So that's the life history. And in trying to work with this organism anywhere, we have to understand where those cysts are and what's driving their dynamics. So that was one of the first things we ever did when we started working up here. Before I get to that, I wanted to cover once again the current systems that we're dealing with. In past talks, I have always just worried about this northern Bering Sea and the Chukchi, and today we're going to go down here into the eastern Bering Sea a little bit. So if you look, you see this red current here that comes along the Aleutians and then comes up along. This is the Alaskan coastal current, sort of a low salinity, actually low nutrient nearshore current that I'll talk about. Notice this orange one off to the west in Russian waters, the Anadyr current. It comes through the Gulf of Anadyr and then up and into Bering Strait. And notice there's other water, the central channel of water that comes up through the Bering Strait. And these are just some of the terms that you're going to hear about. I want to make sure you understand where they are. And as I said, the starting point for this research for us, and maybe it makes sense for this talk too, is here at the cyst stage of this life history. And right now, what you're seeing on the right here is a map of the distribution and abundance of the cysts of Alexandrium catanella in this region. And this is a map that goes from 2018 to 2024. We are still counting the data for 2025. So in this case, it's a composite map of all of these years. And when I first saw the map of 2018, which looks like this, except we didn't have any data down here in the Eastern Bering Sea, I was astounded when the people in my lab gave me the figure. And the reason was that it's enormous compared to anything we've ever seen anywhere, both in area and in density of the cysts. And to show you that, here's this little inset, which is the Gulf of Maine. There's Nova Scotia, Massachusetts, New York is down here, drawn to the same scale in area, drawn to the same scale in density. And you can see how small this feature looks compared to this one. And notice that over here, we have annually recurrent blooms of Alexandrium that close down the shellfish essentially every year in the Gulf of Maine, all driven by these cyst accumulations. And then look, what you have here is something that's 15 or 20 times larger in area and denser. It was extraordinary to see it, and it's been persistent. We've seen it again and again and again, and that's why we can present this composite map. But what you see here are a seedbed up here near Utqiagvik, small but dense. And there's another huge one, the biggest one really, up here near Ledyard Bay and Point Hope, a little bit down into Kotzebue. And then I'm going to skip this for the moment near Nome. You have this other feature down here, which we've newly characterized in the Eastern Bering Sea, which is also very dense and very large. And so you've got, let's say, 4 cyst beds on the US side of the treaty line. And as I'll tell you in a little bit, we firmly believe there's another one over here that we can't sample, but which is actually figuring prominently in the bloom dynamics that we're going to go through. So, but notice the lack of data here. There's no doubt, given that these features end right at this treaty line, that if we were able to go over here, that they would be even bigger than we're showing here. So to give you a summary first, before I give you some real data, we produced this conceptual model a couple of years ago, and I want to go through it again. For this region up here in the Chukchi Sea, we see 2 mechanisms for these Alexandrium blooms. The first one is what we call transported blooms. These are blooms that originate here in the northern Bering Sea, maybe the west, maybe the east, that carry blooms through Bering Strait. And as you see down here in the bottom, these advected or transported blooms, once they get to that stage when they start producing cysts, those cysts fall down to the bottom sediments. And historically, when the water's been very, very cold, what we believe is that there was very little germination of that. This.
You're trying.
I think I did it. So in the historic past, when things were very cold, the cysts were deposited, but it was too cold for them to germinate. These cysts can last for decades, even some people claim 100 years in the sediments. And if conditions aren't right, they just remain in a resting state. They cycle in this resting state. But what has happened in recent years is that these waters up in the Arctic have warmed a lot, the bottom waters as well as the surface waters. And they've reached temperatures now where they can germinate. And these cysts that have been depositing and building up and causing those huge dense seed beds that I showed you are now able to germinate when it's warm enough. And I wrote a paper where we call this the sleeping giant. And for some of these years, it's actually awakening. So what you then have is cysts that germinate, produce vegetative cells, produce a bloom. It's often augmented by these transported blooms, makes new cysts, and then the problem persists. And that's why you can have these annually recurrent blooms, much like we have in the Gulf of Maine. So two mechanisms: transported blooms, blooms initiated locally from that germination. So let's give you some examples. This happened this morning too. Let's see. There we go. So let's give you some examples, first of all, of a bloom initiated from the cyst. Here's some data. Remember, this is the cyst map that I showed you. Here's that big cyst seedbed off of Point Hope in Ledyard Bay. And here are some data from a cruise of the cells up in the surface waters. And notice how this sits directly over this seedbed. Notice also at the time of that cruise, the water temperatures were between 8 and 10 degrees in the area where the cysts were, which is a very, very favorable temperature for germination. And so in this case, you can see there's no evidence of a bloom sort of passing through the strait. This one we believe was a locally initiated bloom. So that was one of those two mechanisms. Now in 2022, the last time I was here, we talked about this bloom. We had just finished a cruise in July and August. And so we didn't have this type of data. But what you see here is an animation of a cruise where it was two back-to-back cruises, actually, from Nome all the way up to here to Kjavik and then back down again, and people got off and new people got on, and then it was up again and back again. So 4 snapshots of this bloom. And you can see by this red bar when we're looking. So when it cycles back to here, you'll see we picked up the bloom first down here near St. Lawrence. You can see it moving through Bering Strait, picking up, intensifying in Kotzebue, and then as we're coming back down, it's disappearing all over almost a 2-month period. So a perfect example of a transported bloom entering the region. But also, if you look in detail, this is now just a snapshot. Notice that when the bloom is very intense here, we see almost no activity of the bloom up here in the region of that big cyst seabed or over here. So we're seeing a transported bloom, but not that locally generated bloom. And why? Well, that answer's here, I believe. If you look, this is the temperature measured in the bottom waters of all these different stations during that cruise. And you can see that it's very cold. And especially if we look in this area near that cyst seedbed, all of these are oranges and almost yellows, 0, 1, 2 degrees. That is very, very cold. Remember, I told you that in 2018, the water was 8 to 10 degrees. So it's much, much colder. And to help you understand what that really means for Alexandrium, a little bit of data here. This is a curve that shows the days it takes until we get cysts to germinate when they're incubated at these different temperatures. And there's cysts in here from the Chukchi and then from the Gulf of Maine and even from Cape Cod. And all of the data points fall on this curve. And you see the red line fits it really beautifully. But that's not a statistical fit. That's an 85-degree day function. You've probably heard of degree days. That's how we determine how cold a winter is. It's a metric that incorporates both temperature and the time you're exposed to that temperature. And what it means is, let's just take that interval where we said we had 8 to 10 degrees in 2018. At 10 degrees, it only takes 8.5 days for those cysts to germinate. But back when we were in the 1 degree range of 2022, it's 85 days to germinate. So it's several months longer just by having differences in the temperature of the bottom waters. And so if you're taking that much time to germinate and it's so cold, the cysts don't germinate, they just stay there, and they're there available for the next year until conditions get better. So here's what I said. Here's the temperatures in 2018. We saw it in 2019 also. Here are the temperatures in the 2022, and there was the difference in time this way between their germination. So in 2022, what we saw was a advected bloom or a transported bloom, but we did not see this this type of a locally initiated bloom. Now, this is something that we want to point out. Where did that bloom in 2022 originate from? One of the instruments we had on board the Norseman when we were taking these cruises was called an IFCB, an imaging flow cytobot. That is like a automated robotic microscope. In this case, it was taking water from the pumps on the ship, and every one of these little blue dots is where the IFCB is collecting data, taking hundreds of thousands of images in a day, and then using machine learning or artificial intelligence to help identify the HAB species and count them. The vectors are ADCP, showing you where the water is moving and how fast. And what you see is we picked up the first part of this bloom over here in the western part of St. Lawrence Island, and the numbers were extremely high already. They're up into the 20,000 cells per liter. We were prepared to issue warnings to all the local communities when we got near 1,000 cells per liter. And so right away, we've got a very dangerous situation. But the point here is more, where did these cells come from? And you can see from these vectors that, first of all, there's nothing coming up here in this Alaskan coastal current. It's just devoid of Alexandrium cells. But everything seems to be coming over here from the western part of the northern Bering Sea. You can see these vectors as they start heading north, and then this jet right here heading up towards Diomede as these populations are rocketed through the strait. So this suggests a source off to the west in the Gulf of Anadyr. We highlight again that the Alaskan coastal current has no Alexandrium cells. And then one thing we did just to sort of confirm our thinking is if we took all of the cells we saw in this particular area on this date, August 8th, and then ran a numerical model backwards, where had the water come from that produced those cells, you can see that as it goes back here, it was in the Gulf of Anadyr. So we have two lines of evidence suggesting that the populations that affect this region have originated in the western part of the Bering Sea. So clearly the northwestern Bering Sea is a major source of these transported cells that deposit cysts into that huge cyst seed bed up here. So in that year, we can take this cartoon and say this is probably the pathway we are the most comfortable with. We did not see this as a pathway that was at least operative then. Okay, so now let's think about the other part of the cyst map. I told you to keep your eyes up to the north, but now this is the whole cyst bed. Now let's just look down here because we've added a lot more samples in the last several years. And what you see are, again, large areas of very high cyst abundances, 4,000, 5,000 cysts per cubic centimeter and higher, very much like we saw up north. And to show you that it really does matter, here are some data from 22, 24, and 25, where we used that IFCB on two different cruises. And the size and of these circles tells you how many cells were being observed by that instrument. And you can see that very close to St. Paul Island, basically right in here above this seabed, we are seeing a bloom. And to accentuate that further, in 2024, and this we'll hear more about this from Patrick, there was a die-off on St. Paul Island of the northern fur seals that was, we believe, conclusively linked to saxitoxin. And you can see from what I just showed you that in 2024, there were bloom cells here very much near this seabed. And then much more recently, in this year, we have had another die-off and another set of toxin measurements in St. George Island, where seabirds, marine mammals, shellfish, and fish samples were tested and found to contain elevated levels of PSCs. So this area that we've just highlighted with the cis seabed is clearly functioning with major blooms. And we're trying now to figure out where, is this the same situation where you have two mechanisms, where there's these locally initiated blooms here near the Pribilofs from the cis seabed we've characterized, or is it also happening that there are transported blooms? We know that the Gulf of Alaska has a lot of paralytic shellfish poisoning problems. Are there blooms being carried along the Aleutians and then through some of the passes that are contributing to that, much like we had two mechanisms up here? So we don't know the answer to that yet, but we're trying to get support to look into it. And the thing that we want to emphasize, though, is that it's not just an academic exercise to figure out what's causing blooms in this really remote area, but we actually do believe there are hydrographic pathways that can take these blooms up and into this region. We didn't see it in 2022, but we know it can happen. And here's the evidence for that. What we've done is put passive particles here in the Gulf of Anadyr, and a number of them here where that other cis seabed is. And this is each one of these is a month, and this goes into August to September and then October. And what you see with just the currents for that particular year, this was written as if for 2022. You can see that, as we saw before, the Anadyr pathway is there, that in a relatively short period of time, passive particles from here make it up and into Bering Strait, and you can see through time they go well up into the Chukchi and beyond. But notice that the water also, had there been a bloom and cells here, comes up this eastern pathway into Bering Strait and also travels up here. So basically, both pathways are valid for transport of these cells from these bloom initiation zones. So what we've added is a new cis seabed and a new potential transport pathway, which we think and know wasn't operative when we were there in 22, but we think it can be in other years. So again, hold on, there we go. So let's look at one more thing before we finish here. People in Nome, right away when I showed this map, Gay Sheffield saw this and said, what is that? And what you see here was a bullseye of cis right near Nome. And if we go back, look at 2022, we didn't see anything there when we did our cis mapping down here. 2023, there it is. And it's not just a single data point outlining that area, that hotspot, it's quite a few. And this would have been the year following that big 2022 event. And then, but look here in 2024, now it's getting smaller, diminishing, and we're counting the 2025 data now. My guess is it will be even smaller. And the reason I say this, we've seen this before in other parts of the country, in the Gulf of Maine, where we have a big event that drops cis, but they're dropped into areas that are not necessarily accumulation zones, where storms and waves and currents can resuspend them and gradually move them into deeper areas with calmer waters where they will deposit. So we think the seabeds that we've been highlighting, those three big ones, are persistent features. And then you have these occasional features like the one near Nome. So the good news is that at least Nome doesn't have this huge seabed forming or formed on its doorstep. It looks like it's going away, and you have to stay tuned to see if that bears out. Okay, so what I've pointed out is that now we've got transported blooms and blooms initiated locally, potentially in both parts of the northern Alaska and in eastern part of the Bering Sea. So again, I don't know why this would be a green, it would be easier to see, but notice that in 2018, we saw a locally initiated bloom. In 2019, I didn't go through that. We saw both a transported bloom and a locally initiated bloom up north. We're talking in the Chukchi Sea area. 2022, you saw this. This was a transported bloom with no local initiation. 23, we saw a transported bloom, no local initiation. But notice this, 2024 and 2025, nothing, neither one. We're still, as you'll hear from Patrick, a little mystified because as he'll point out, in 2025, they found walrus having quite a bit of PSP toxins in their feces. But we, in several cruises, did not see any of the Alexander cells that might have caused that. So that remains a bit of a mystery. So why are we seeing that kind of variability? Well, the answers, I think, lie in the bottom temperatures. So now here's the time series of Ledyard Bay bottom temperatures through May, June, July, August, September. This is the bloom season. And you can see in this 20-some year record, which ones are the cold years and which ones are the warm years. And notice here's 2018 and 2019, both very warm years. This is the bottom water. And notice here's 22, 24, and 25 are sort of in the lower part of this whole spectrum here. And so what we can do is take that function I showed you earlier about degree days, and instead of just saying how many days is going to take to germinate, let's turn it into a date. So these are just, this is July 23rd and August 2nd. And now we do it for all of these years using the GLORIES data set, the archived runs to show you what bottom temperature would have been in this region. And what you see is these are the years that were warm, and the germination would have been in sort of mid-July. And the years that were much colder, you can see the germination is roughly a month later. And we think that that month is critical in terms of how many cells are able, how fast they can divide in the surface waters and how much time. It's like planting your tomatoes late in the season. You may get some plants, but they're not necessarily going to be very big ones. So that, what I'm pointing out though, is the interannual variability in temperature is really dramatic, and it's having a huge effect on those locally initiated blooms. The transported ones, I think, are a different story. The waters are warmer down there, and I think those are going to continue. So to answer that last little bit, we've also done the same thing. I just went through to look at these dates of the degree days. Over here, we put a box off of this region, right near where the St. Paul Island is and that seabed is, and here another box. And we put them here because one of these is in this area that's called the cold pool. And you can see this is 2009, a really cold year. That water down there in that region is -1, -2 degrees. And notice a warm year down there, 2018, the water is much warmer up here in the 4, 5, 6 degree range. And then if you go through that same analysis, what you see is in the year when it was really cold, that the germination was August, September, really late in the year, too late to grow a bloom. And the years that are warmer, much, much earlier. Now we're talking June, July. And remember that mortality of seals and so forth on St. Paul, St. George, those are happening in July and August down in this region. So it all tends to fit together nicely. So with that, actually, this was from a different talk. I'm going to leave that out. I'm going to give you some final thoughts. We have characterized at least 3 major seabeds in the US waters of the Alaskan Arctic, and that stretches from the eastern Bering Sea to the entrance to the Beaufort Sea. We hypothesize another one in Russian waters of the western Bering Sea, but you also saw a smaller cisbed south of Nome first appeared, but has since diminished in size, and it may continue. But the scale of the overall cisbed is enormous, and with multiple sites for toxic bloom initiation throughout the region. And our analyses of interannual temperature variability and using particle transport modeling are giving us insights into bloom timing and transport. And we now, I think this is an important statement. It's now clear to me, at least, that these Arctic cyst beds operate independently with dynamics varying dramatically from location to location and year to year because of those differences in bottom temperatures. So one cyst bed might be warm enough to produce cysts, another one's too cold. They really are functioning independently. And the transport pathways are also important. Some years they're strong and are taking a bloom, and other years they may be there, but there's no bloom to transport. And the final point being that transported blooms can affect communities far from these seed beds. So with the exception of that eastern Bering Sea cyst bed, bottom temperatures are often too cold for germination up here. And that's what I think is leading to the increases in cyst abundance because of that imbalance between inputs and losses from germination. And I think that's why the cyst seed beds are so dense here. In warmer years, these high bottom temperatures do lead to these locally initiated blooms. And finally, I think the transported blooms are the largest threat to humans and ecosystems in the Bering Strait, Kotzebue Sound regions, while the locally initiated blooms are the main concern from Ledyard Bay up to the Utqiagvik. I don't know what to say about Eastern Bering Sea right now. I think there's both the transported and locally generated possibilities, and we don't know which are more important. So I've left that out. So with that, I'm going to stop and hand it over to Patrick.
And save your questions for the end. Patrick's done.
Thanks, Don, for that great overview. So I'm going to be just going over, so we discussed kind of Alexandrium and where the bloom dynamics in the Alaskan Arctic and Bering Sea regions. And now I kind of want to talk about the toxins that they produce. We mostly will focus on Alexandrium and paralytic shellfish toxins or saxitoxins, but we'll also touch on domainic acids. So as Don pointed out, there's various pathways in which the toxins that are produced by these algae can enter into the food web. And so one thing that I think is important to just identify is that the mechanisms of toxicity differ between domainic acid, so that is the neurotoxin that causes amnesiac shellfish poisoning that Don mentioned with memory impairment. That is what we call a excitotoxin. So its mechanism of toxicity is actually overstimulating the central nervous system. And so when a marine mammal like a sea lion is actually impacted by domainic acid poisoning, these are the types of symptoms that we'll exhibit. It's going to be having seizures, memory impairment. If you ever see something on the news where sea lion is on top of a car or in some weird area that they're not supposed to be in, that could be a symptom of domainic acid poisoning. Now, saxitoxin, on the other hand, which is produced by the Alexandrium, is a paralytic. And so the mechanism of toxicity is a bit different. So this is some work done by Kathi looking at larval fish exposure to saxitoxin. So in a control where there's no saxitoxin in the water, poke a larval fish, instinctively it'll react to that stimulus and swim away. But if this larval fish is exposed to saxitoxin, it is pretty much shutting down its central nervous system. So you're going to get, for humans, if you're exposed to saxitoxin, one of the first symptoms is tingling in the lips. You might start to feel paralysis in your limbs. If you're exposed to a high enough dose, it can cause respiratory arrest or eventually cause your body to shut down and stop breathing. So really, your only hope for surviving is being put on a respirator. So keeping those mechanisms of toxicity in mind, I wanted to just share the wealth of samples that we have collected in terms of food web samples where we tested for both of these toxins in various food web samples, ranging from zooplankton, fish, clams, worms, and then eventually we expanded to other invertebrates. And this is just a picture of myself and Evie. That's actually the 2022 cruise where we were part of that team that detected that massive bloom that Don just talked about. And then that's Emily Bowers, who's our awesome lab manager in the Warren West lab. So first we'll start off with domainic acid. So just to orient you to what these figures kind of show, you can see the different icons represent a different food web sample we collected on one of the various cruises of opportunity that is collaborative on this research. And so the color of the icon represents the amount of domainic acid that we measured in it. So if it's white, that means below detectable limits, or BDL, so the assay wasn't able to detect that toxin. If it's yellow, we would term that as low, having low levels of that toxin. Orange is about half the regulatory limit, so we would call that a moderate amount of toxin. And then for red, those are animals that had domainic acid at or above the seafood regulatory limit. And so looking at 2019, we had really good sampling coverage. And there was also a big, massive bloom of Alexandrium, but this is DA. So what we see here is pretty much low to BDL levels of domainic acid. And so we see a lot of white icons and yellow in all the different types of samples we collected and tested. 2020, we were only able to get a limited amount of samples due to the pandemic, but you can see it's kind of a similar trend where we see below detectable levels of domainic acid or kind of low levels. 2021, again, another pandemic year, so not a ton of samples, although we were still able to get some. And again, a similar result where we have low levels to below detectable limits of the assay. And in 2022, even with extensive sampling, we have great coverage. And throughout all of those samples, we still see a similar trend of background levels of domainic acid in the food web, or BDL, right? So those white icons. And in 2023, again, another year with good coverage, we still see a similar result where we see either low levels to below detectable limits of domainic acid in the food web. So the results here, the summary from these data show that domainic acid concentrations were low to non-detectable in all layers of the food web, whether it's zooplankton, invertebrates. It was either BDL or pretty low. We did not measure any samples that got near the regulatory limit. So this is good. So what this means is that domainic acid is present kind of in background levels. So the pseudonychia are present, but they're not there in high enough levels to be a concern right now. But with saxitoxin, that is a different story. So saxitoxin is a higher risk in Arctic food webs than domainic acid, and you'll see why. So again, this is a very similar type of figure, except for domainic acid. Now we're showing you saxitoxin. So here, if we look at those same years and the similar data presentation, you can see we collected this is a good year of coverage for sampling the food web. And you can see clams and a zooplankton sample that reached the seafood regulatory limit or above for saxitoxin. And then we also see not too many white icon. So what that means is the prevalency is pretty high. So even if we collect a sample, most likely you get a positive result for saxitoxin above that BDL. So it's still in the environment. And then in those certain areas with those clams, we're seeing those really high levels accumulate. 2020, again, pandemic year, not a ton of samples, but even though we had limited samples, we still were able to detect saxitoxin at or above the seafood regulatory limit throughout the Chukchi and in the Bering Strait region. 2021, again, another pandemic year, we got a bit better coverage in the Bering Sea. And again, even with relatively limited sampling capabilities, we were still able to measure saxitoxin at or above the regulatory limit. We have those hot clams. And in 2022, during that massive bloom, and we were able to collect a little bit wider variety of food web samples. We were incorporating anemones, well, I guess that's the other food web sample that is kind of note, because those also were able to contain saxitoxin at really high levels in that red category. And again, we see the similar culprits that we see in other years. We see clams at the seafood regulatory limit. And I believe we also had some gastropods too. They may be hidden there though. And then in 2023, another bloom year, we see similar results with saxitoxin really accumulating in clams, in worms, and also sea anemones at moderate to high levels. And again, the prevalency is pretty high. So it's present in the environment, and in those animals, they're present at high concentrations. So these results point towards domainic acid is not a current threat to the food web here in northern and western Alaska. Domainic acid, though, is a big problem off of the US contiguous West Coast, so California and Washington. Every summer, you may hear on the news of sea lions and dolphins stranding with seizures, like I showed in that video. So they are really dealing with domainic acid poisoning that accumulates in plankton feeding fish. And then sea lions and dolphins are definitely exposed to those high levels. Whereas here in Alaska, saxitoxin is really the main concern. So we see these really accumulating in high levels in the filter feeding benthic invertebrates like clams. And that makes us believe that benthic feeders like walruses are kind of the most susceptible to being exposed to saxitoxin's effects. So, so there were 2 summers that Don talked about where we had well-characterized, really good sampling of the blooms. That was in the summer of 2019 and in the summer of 2022 in the Bering Strait and Alaskan Arctic region. So that really posed a question to us. And then also knowing that saxitoxin is accumulating in some of the preferred prey of walruses, it begs the question, kind of what are the exposure risks then for these paralytic shellfish toxins to walruses during these massive Alexandrian blooms? And so what we can do with bloom data and then measuring the toxins in the walrus prey is we can kind of build models that help estimate how much toxin is transferred from the Alexandrium up to the food of the walrus, and then up to the walruses themselves. And so luckily we do know, and we're able to kind of estimate the diet and foraging ecology of different benthic invertebrates. So those arrows are just pointing towards which animals consume those items. So Clams typically tend to eat the cells and are able to also consume cysts. Worms are deposit feeders that live in the sediment. We think they would be mostly exposed through potentially consuming cysts. And then we know another prey item of walruses are snails, and they actually like to eat clams, which, as we just showed, is a really good vector of these of saxitoxin to different trophic levels. And so if we know bloom conditions, like how many cells are in the water, how many cysts are in the sediments, and the toxicity of those, we can estimate that. We can estimate the amount of toxin then that moves into these benthic invertebrates based on these trophic pathways. And then we do the same thing for estimating toxin exposure to walruses through their diet. So we know, luckily, there's a lot of literature out on walrus metabolism, caloric requirements, preferred prey. So we combine that literature with the data we have looking at the saxitoxin in their prey, and we can build models that estimate their exposure via their diet during these blooms. And so what I'll be showing you, so what those models kind of estimate are what's called a daily oral PST dose. So that is a number we get, and all that means is, okay, based on where a walrus is foraging, based on their caloric requirements, their preferred prey, the amount of toxin in their prey, how much toxin would a walrus be exposed to during one day of feeding? So to build these models, we collected Alexandrium data, those vegetative cells, the cyst density data, and PSTs by either collecting and filtering water, which I'm doing in that picture in the upper left-hand corner there, to estimate cell densities, and then also we can measure the toxin in those cells. And then also we use the image flow cytobot, which in a nutshell can give us real-time bloom data by estimating how many cells are in the water while a ship is underway. And so we combine those data with the cyst data that we're able to obtain through deploying what are called Van Veen grabs. So I like to describe these as, if you think of the arcade game with the claw, and you dispense the claw down to go get your prize in that arcade machine. Instead of a claw, you're dispensing a big scoop, and the prize we're trying to get is a huge batch of sediments. And so we bring those sediments to the surface. We'll take a sediment sample that we ship off to Don and Evie at Woods Hole, and they'll go count the cysts in the sediment. And then we'll also filter those sediments and collect the invertebrates and test those invertebrates for PSTs. And so we have these paired data at each of those stations. That's what those figures were showing. So in 2019, that's where, in 2022, where we collected the Alexandrium data. And then at those same stations, we also tried to collect invertebrates. So we had this paired Alexandrium and invertebrate PST data set to build these models. And so at each of those stations, we use the amount of toxin we measured in the walrus prey, to estimate one of those daily oral walrus PST doses. And so before I show you kind of the results from those models, I just want to orient you to what these colors represent. So we don't know exactly right now how much toxin or saxitoxin or PST, how much of it takes to impact the health of walruses. So we don't know if it's exposed to a dose of 5 micrograms of saxitoxin equivalent per kilogram, or if 20, if those are high or low estimates. So what we had to do is look through the literature to see what has been shown to impact the health of other animals. And so what we found is doses in this blue color range really weren't associated with any significant health impacts. So there wasn't any die-off events associated with those doses. So we assumed that if walruses were exposed to doses in this range, most likely this would represent a low health risk. But as we get up into the yellow colors, this 11.6 dose, that is associated with a known mortality event of humpback whales that were known to feed on saxitoxin contaminated fish. And so, however, that was kind of an opportunistic event. The dose estimate was preliminary, but it was a confirmed death event due to saxitoxin. So as we go kind of higher up in our dosage, they've done controlled feeding studies of PSTs to dogs, and they found that, and so these are well controlled, they know exactly how much toxin they're exposing the dogs to, and they found that at this dosage, it would kill about 50% of the dogs. So we would hypothesize that, okay, if walruses were exposed to these types of doses, then it would probably pose a moderate health risk to walrus. And then that red color, this 80 micrograms of saxitoxin equivalent per kilogram of body mass, that is a very conservative dose for a lethal dose to humans. So if a human is exposed to this amount of toxin, they most likely die. So we would assume if walruses were exposed to that amount of toxin, these types of doses would probably represent a really high health risk to walruses. So that is how we developed this color ramp, this scale, and this walrus health risk index. Okay, so now looking at, this is a what we call a walrus PST dose map. So at each of those circles, here, these gray circles. So those are each a station where we sampled Alexandrium and benthic invertebrates. We measured the toxin in the invertebrates. And based on that measured toxin value, we estimated how much toxin walruses would be exposed to if they were foraging in that area for a day. And these stations were sampled from August 5th to the 22nd in 2019. You can see that a lot of the doses were above that moderate health risk threshold. We even have some of those dark orange colors and even a few stations. So 9% of those 53 stations actually had a really high health risk dose to walrus. So this right now is just kind of a tool to estimate potential risk. So it really helps to know if walruses, where the walruses were, or where some walruses were. And so USGS at that same time frame was actually tagging walruses out on Point Lay. And so that's what you're seeing here in that inset map. It's the same dose map I just showed you, just a zoomed in version on Ledger Bay. And so that is tracking data from 26 walruses. So it's not a ton of walruses. And
those are their locations.
Yes.
That's what those tags are doing.
yeah, but each of those circles is a satellite location of a walrus. So you can see most of them stay close to the coast. There was one walrus that traversed out into Ledger Bay, but that's not a population level movement pattern. The more normal pattern is for walruses to go take a trip out to Hannah Shoal, because that's a really rich foraging area for them. And so what we found is that Ledger Bay, which had almost all of those really high risk doses, we sampled a station at Ledger Bay for those invertebrates. And on the same day, the first walrus was tagged at Point Lay. So what that just means is that these doses were estimated, or they're based on toxin data measured in walrus prey at the time that walruses were actually close to the area. So potentially walruses could have been exposed to these types of doses. Now switching to the 2022 bloom, we had a bit more spatial coverage, a bit more stations, but you see, again, lower overall dose estimates. So if we look at that breakdown for the 61 stations, we see a lower percentage of the doses were above that moderate health risk threshold, but it's still, a majority of the doses are still relatively high. But we don't have any of those red colors, right? So those high health risk doses, which is good. Again, this is just a tool to look at potential exposure, but where are the walruses? So again, we're looking at kind of Point Lay, and that haul out in USGS had satellite images that were used to estimate the number of walruses at the Point Lay haul out. Those data started August 15th through October 10th. And we see on August 22nd, walruses start to occupy the Point Lay haul out. But on August 29th, we see significant numbers start arriving and using that Point Lay haul out. And Ledger Bay, that invertebrate sampling that we did and those moderate, those yellow, light orange doses, those are based on invertebrates sampled for saxitoxin during August 2nd to the 10th. So just a couple weeks before walruses start to arrive at Point Lay. And then I'll also make note, Gabe pointed out that we have these dark orange colors. So these are kind of the highest doses that we found in 2022. And walruses were found to wash ashore on the Seward Peninsula.
North Shore,
North Shore, where's that?
there.
Right here?
No, Wales to Cape Aspenberg.
Okay.
That flat top. Oh.
They won't see this, but
Yeah, no, that's fine. Okay, from here to here. So we don't know if those walruses were, those walruses were documented in August, I believe.
Yeah.
And so that would be after the bloom, or, yeah, that would be either during the tail end of the bloom. We don't know if those walruses had paralytic shellfish poisoning, but it's just an interesting thing to note that there was documentation of walruses washing up here. So all that to say is that during these blooms, these doses, walruses are around certain areas. So there is possible exposure. One bright note, though, is that you'll see in Hannah Shoal, those doses actually decreased. So the Hannah Shoal area tends to have lower doses compared to like really high risk areas like Ledger Bay. So potentially the areas that walruses like to use, there could be lower exposure when a big bloom is happening. Although if you remember in 2019, those doses for that area were still above that moderate health risk threshold. So it could still be potentially hazardous. So summary of the exposure risk data for walruses is that those models estimated high PST doses for walruses in 2019 and 2022. These doses, if walruses are feeding in those areas, they would probably be exposed to levels that could cause some kind of negative health impact. We don't exactly know what those are though right now. We are trying to address that though. Walruses were at the Point Lay haul out during or soon after invertebrate sampling during both of the blooms in 2019 and 2022. So we would kind of say that Ledger Bay is a high-risk area for exposure for walruses if there's a bloom that happens in that area. And then we can use these models to actually assess exposure risks during future blooms. So if we do know bloom densities, cyst densities, and we can estimate toxicities, we can build those models based on this work here. Or we can simulate different bloom scenarios and see what kind of exposure risks walrus would be exposed to. So those models are really great for looking at, potential exposure risks for PSTs during blooms. But here, Don alluded to a little bit earlier in his talk, I'm going to talk about some results we're finding from observed algal toxin risks. So we never, part of that modeling, we didn't actually sample any walruses for saxitoxin or PSTs. Here we're including some of those data from walrus cruises we've been a part of. So when I talk about the walrus cruises, these are cruises that are led by Irina Trukhanova at U.S. Fish and Wildlife Service and Bill Beatty at USGS. These cruises are conducted in the Bering Strait, Chukchi Sea areas during a pretty extensive time period. It's about a 4-week period in June. And the objective for Irina and Bill are to conduct mark recapture and age structure surveys to look at walrus abundance and population trends. Yes, Jay?
I'm just gonna say, we've hit we're coming up at 20 till 8. I'm just wanting to give the audience a chance to know how much time do you think you will need to, because this will be interesting. I'm interested. We're talking about walruses, but in case people are having to figure out what they're doing, do you know about how much more time you have?
Probably like 15.
15 minutes.
10,
Okay,
15.
that's it. Keep going. Keep
Okay.
going. Thank you.
Yep. So point of this cruise, collect biopsy samples to look at walrus abundance and population trends. They contacted Kathi. Hey, we have these cruises. Does this interest you for potential HAB sampling? And it did. So we sent out HAB samplers in 2023. Emily Bowers from Kathy's lab. I went out in 2024, but there's an asterisk there because we got stuck in ice for 2 weeks. So we didn't get a ton of sampling near walruses. This past June, Madeline Kowalik went out, a local community member here at NOAA, and she was awesome at getting all of the HAB samples that we requested from her, and we trained her over a 3-hour Zoom. So she was great, did a lot of great work. So the really unique thing about this opportunity is that we're sampling for HAB activity in areas where walruses are actively present foraging. And so these walruses are they're sampling along the sea ice. And so we also get to see really awesome wildlife like walruses and polar bears. But the samples we do try and collect are water samples to look at cell densities of Alexandrium, or sediments to look at cyst densities, invertebrates to look at the toxin content of walrus prey. And then here, this is kind of the cool part of this collection is we look at toxin and walrus scat that we'll go and collect out on the sea ice flows. And they try to target fresh walrus scat. And so we're looking at benthic invertebrates, testing invertebrates for domainic acid, again, where walruses are present. We see similar results to what I showed you earlier, very low to below detectable limits in each year. And if we look at the walrus scat, we see similar results. So pretty low domainic acid and walrus scat, so low exposure. Well, that's interesting. But the summary here with domainic acid is that it's present in food web. in low concentrations, kind of background concentrations. And we think that based on that and the low concentrations in the walrus scat, that these domainic acid levels are not a significant health threat to walruses. Moving on to saxitoxins. So these are looking at invertebrates, and then those purple circles are cyst densities. You can see in 2023, saxitoxin is a different story than domainic acid. So we see saxitoxin at or above seafood regulatory limits in clams and even an anemone. And we see them near really high cyst density areas. If we look at 2024, the only thing I want to note here is that we were completely surrounded by sea ice. And so while we were collecting these samples, I didn't think we'd find anything too out of the norm, but we actually detected saxitoxin at or above the seafood regulatory limit in a snail. So dense sea ice does not preclude high levels of saxitoxin. At least this one little bit of evidence points towards that. 2025, we see kind of similar results to 2023. We see high levels of saxitoxin in these invertebrates, kind of in similar areas we see in 2023. So if we look at these results and compare them to the walrus scat is on the right figure, we see in areas with high saxitoxin in clams and sea anemones and cyst densities, we see high levels of saxitoxin in the scat. But we also see low saxitoxin concentrations in areas with low cyst densities. And that also corresponds to low saxitoxin in the walrus scat. 2025, this is some pretty interesting results that Don alluded to earlier. So we didn't detect an Alexandrian bloom, but we sampled these invertebrates in June, and clearly you can see very high levels of saxitoxin in clams and anemones, gastropods, a worm, and that corresponds to really high levels of saxitoxin in the scat. And so we think there could be kind of two patterns we're seeing with these data. In the green circles, we're seeing areas with high cyst densities are leading to high saxitoxin in the prey, leading to high saxitoxin in the walrus scat. And then in the blue, these colors are not correct, but we see low cyst densities that correspond to below detectable limits or low levels of saxitoxin in the prey and low levels of saxitoxin in the walrus scat. So really we're thinking cyst density in combination with sea ice. So sea ice is kind of, the extent of sea ice seems to dictate where walruses will be feeding, maybe drivers of walrus saxitoxin exposure. We're not saying that walruses are eating cyst. We're saying the high cyst densities seem to be correlated with high saxitoxin in the prey, which walruses are consuming and then being exposed to the saxitoxin that way. Okay, and then this is the last thing I'll go over is the die-off event of northern fur seals in the Southeast Bering Sea. So there was on August 18th, about 10 dead northern fur seals along with a bunch of fish were found on Benson Beach on St. Paul Island. This was a monumental effort by tribal environmental staff, community members on St. Paul Island, and then veterinarians and all types of researchers. But staff on island collected and froze 6 northern fur seal carcasses, as well as some fish. At the same time of that die-off, we had just opportunistically had 2 different research cruises that were collecting some Alexandrium data for us, the Healy and the Dyson. The tracks represent IFCB data for those cruises, and the shapes represent stations where cysts and or food web samples were collected for saxitoxin testing. And so when we looked at the saxitoxin levels in the tissues of the northern fur seals, which I should note, these northern fur seals were found in a pretty good body condition. And they also, veterinarians did necropsies, didn't find any overt cause of death, tested for avian influenza, that was negative. But we did see relatively high levels of saxitoxin in the feces. And then kind of the smoking gun for us was this urine saxitoxin concentration of above the seafood regulatory limit of 800 nanograms per mil. So urine is where the toxin is excreted from the body. So this confirms exposure to the toxin. And so you pair that with, in July, we see bloom densities of Alexandrium in July and in August. The saxitoxin we measured in those food web samples, prevalency was almost 100% in all the different sample types. And then, as Don alluded to earlier, there's these massive cyst beds here. All this data together pointed towards a poisoning event of these northern fur seals. And then this is just showing some particle backtracking data. So the magenta points are where the animals potentially were washed up on Benson Beach. And if you backtrack the particles, we can see that they potentially originated in the area where we see those blooms, the high cyst densities, and the saxitoxin in the different food web samples. So this was the first study to combine Alexandrian bloom and cyst data, environmental saxitoxin concentrations, the multiple layers of food web, and some oceanography data to define a saxitoxin poisoning event in a marine mammal species. And we just want to give a special shout out to community members on St. Paul Island. It was a huge effort by all involved. Very briefly, take-home messages. It's really paralytic shellfish toxins that are a threat to the health of marine mammals in Western and Northern Alaskan waters. We are still working on what doses will lead to health impacts of marine mammals. And we are beginning a project that focuses on algal toxin impacts to marine subsistence resources. So more to come. So thank you. Sorry for
No, it's all good.
Yeah.
Yeah, thank you both. And before we start questions, this is the time for, while you're thinking of your questions, this is the time for the Straight Science audience to show some love in the chat for Don Anderson and Patrick Charapata. They've come a long way and have taken the time out of their day to give us a very thorough Straight Science on harmful algal bloom activity in Western and Northern Alaska. So show some love in the chat, and then we'll lead off with questions. And I think you guys will start. Let me check the computer here, see if we've got anything going in the chat. You got some kudos, but a question. I'll start off with a question, and that is the cyst bed for that Don Anderson showed. You might want to show your slide on that. The cyst bed off of Nome, is that left over from 2022, do you think? That was the big year of the, and that's right off our coastline here.
trying to figure out how to escape out of this. There we go.
Those are your Nome cysts, because that kind of caught my attention there.
Yeah, it's a very,
Or are we just still learning about it? So we're just waiting and more to follow, kind of thing.
the answer is the latter, in the sense that we don't know very much about it. That the problem we have
This one,
is
right?
that, yes, this little spot there. And I guess I should go back further because what we want to see is
There you
this.
go.
And this this cruise in 2022 was actually, I don't know, that's the critical answer. I don't know when this Eastern Bering Sea cruise happened relative to when the bloom was occurring in 2022, which was July and August. So I can't directly answer your question. Somebody in my lab could, but not me. So one thought is that this occurred before this map was before that bloom occurred, and therefore there wasn't really anything to drop here in 2022. That would not be a very good explanation. 2023, we had limited cruises, but we did pick up an event in Bering Strait, but not what we didn't sample down here. So we don't know if there was a bloom in this lower area in that year. My thinking is that in fact, yes, it was related to the cells and the bloom in the St. Lawrence area. If I can find another figure, it will help me explain that, why I would say that. I'm not sure it's in this presentation, but this one will.
That's all right.
If
That's
we
all right.
looked at the IFCB data, this simulation here almost shows it. Well, this figure. If you look at the fact that when we ran the IFCB, this one's going to cycle through, but if you look at the beginning of this, there were cells moving to the west of St. Lawrence, but also here to the east. It's blue there, but in fact, there were cells making this transit. And then the boat never really came back here. So it could well be that in 2022, we had cells in this region that dropped that showed up as 2023 as that hotspot. That would be my most likely guess. Given that the flow in Bering Strait is all to the north, it doesn't see. There are a few cells that were here a little bit south of the strait, right there, and that's right where the cysts were found. So that's my best guess, and that they got dropped in, but they're being eroded away, and those cysts are ending up somewhere else.
All right, thank you very much. We have two, Claudia, your hand is up, and we also have a related question to this cyst bed in the chat. Claudia, are you is yours a cyst bed question?
No, not exactly a system. Yeah.
Deanna Hacker has a cyst bed question that might be really quick for Don to answer, so it doesn't have to change his slides. But, and it actually says question for Patrick, but I think it's for Don. It says, regarding that cyst bed off of Nome, are there any studies on how this could affect the king crab fishery and or the safe consumption of king crab?
The quick answer is no. We've never, I would love to try to start looking at king crabs in the context of all of this, but it has never been something that we've gotten data on or had collaborators come up to us and say, hey, let's try to match our catch data to your cyst or cell data. And I also have never heard of any toxin measurements out of king crab. What we know for crabs is that the toxin ends up being in the organs and so forth, not in the flesh. And so I don't know whether anybody measures that. And so the answer is no, but we'd love to try to get more information there. I think that would be a really.
We have a processing plant right here in Nome, and we have a commercial king crab harvest. We also have which might be, because everything about HABs is different up in the north, and so we have almost complete utilization of a crab for consumption, including the gills and the just about every part of it. So it might be something to be of that would be of great interest in the region, I think, both for subsistence and
well worth looking at, because I'm worried about the levels that you might find in those other tissues
right.
of the king crab.
Claudia, hold on one more sec.
So my
Question from the audience.
does saxitoxin bioaccumulate in crabs?
It's bioaccumulate is a tough word. I don't consider it bioaccumulating in the shellfish. My thinking of bioaccumulation is we have other toxins that are fat soluble and goes from, like say it's a fish, from an algae to a little herbivore, and from the herbivore to a carnivore, and the carnivore to a bigger carnivore. And that's sort of a bioaccumulation like you might get with a pollutant. At least that's my view. I may be wrong, but that's not what we see in a shellfish. It's just gradual accumulation because it just keeps filtering and filtering. A little bit is lost from that shellfish, but not at the rate that it's accumulating things. A crab probably is the same thing. It's eating clams, let's say, getting a toxic dose. It's that toxin is probably not just in its stomach. I would think that some of it's being bound to tissues. You know that happens in shellfish, that there's certain parts of the shellfish that actually bind the toxin and hang on to it. And that's why some of these clams can hold toxin for over a year. It's not in their stomachs, it's in their other tissues. My thinking is that's the same for crabs, but I don't know. There's probably some studies, but that suggests another thing for us to do in our new project, is to try to look at that. But if you've got,
Patrick.
yeah, if you've got some processing plants, and especially to answer that question, if the crabs came from areas where we have a lot of cysts, and they're, and potentially then, like Patrick was seeing, clams and such that are toxic, that would be really interesting to get measurements of those, the parts of the crab that are sometimes thrown away.
Thank you. Good question. So, and thank you for your answer. So Claudia and then Charlie, and then we'll go to the chat. There's also a question in the chat. Go for it, Claudia. Thank you for being so patient.
Yeah, sure. So my question is, what is known about the role of saxitoxin in the biology of the algae cell itself? Is it just a metabolic byproduct? And if so, do they just produce it proportionally to their metabolic rate, or are there certain ocean conditions where they might make more or less of the stuff?
Well, the first part of that question, we don't know. After all of these many years of study, there are many studies that show that certain zooplankton are affected, and in fact, one more recent line of studies suggests that the presence of certain copepods or zooplankton and some exudates that they produce make the algae produce much more toxin. And that's the first sort of line of evidence that I've ever seen that starts to suggest it has some defensive role. But it's, that it still remains a very open question. The second part of your question, though, was does the toxin vary? And indeed it does. We can in the lab, by growing the cells under different nutrient conditions, can vary toxicity at least by an order of magnitude. So different nutrient limitations can produce weakly toxic cells or really extremely toxic cells. So yes, you can have that kind of variability, but I think that level of variability is dwarfed by what people are reporting about the effect of this material called copepotamide, which seems to really stimulate Alexandrium cells to produce a lot of toxin. And I should say that both Mike Brosnahan, who's here in the audience with me, and I have tried to see if we could stimulate some of our Alexandrium to produce it, and we haven't succeeded. but there are many papers in the literature that suggest that other strains respond to that. So right now, I would say it's a reasonable guess that in some cases it's a defensive mechanism, but other than that, we don't know much.
Good question, Claudia. Thank you.
Yeah, thanks.
And Charlie, you've been very patient. Go ahead.
No, thanks. I guess I had a comment before my question. So the state PSP lab requires annual testing of shellfish in Alaska. So the crab in Norton Sound have been tested and have passed. The what I understand from that is that some PSP does accumulate in the hepatopancreas, the organ that produces digestive juices. It's the yellow crab butter, some people refer to it as. And that's where, if you were going to get PSP, it would be most likely. It doesn't accumulate in the muscle. So I think trying to put people's mind at ease a little bit so that does occur, but it's generally not in the portion that's sold, at least. I know lots of people eat the crab butter, but that's where I'd be worried about it. I guess my question was the symptoms for PSP and botulism are very similar, and occasionally in the Norton Sound region there are cases of botulism poisoning, and I wonder if that's possibly PSP and not botulism. And I've been around eating things like sea strawberries and other strange invertebrates that are bottom dwelling, and people tell me, don't eat too much of that, your eyes will cross. Don't eat too much of that, your lips will tingle. I think there's a lot of things that might be picking that up. And I just wondered if you had any idea about botulism versus PSP symptoms.
Okay, yeah.
Let me just start here and then Patrick can make a comment. The first is that I agree with what you're saying about the crabs. As I said, we have never, it's not just crabs, it's in fish and other animals. The mussels are generally toxin free, and when we worry about toxin in a crab, it would be in the hepatopancreas, in other types of tissues, often the ones that are not eaten, but in other parts of the state, clearly they are eaten. You said that Norton Sound has been tested, but right now, except for that little cyst accumulation I showed you from 23, there really aren't a lot of cysts or a lot of cells that we know of in Norton Sound. I'd love to see data on crabs from some other parts of the Bering Sea, let's say. And if anyone has that, that would be nice to hear of. But then about the symptomology, I'll let Patrick have some thoughts, but it's not unlike we hear people say, well, when people are drunk, they can be unsteady, they can feel disoriented, and some of the symptoms for saxitoxin or PSP poisoning resemble that. And I'm quite sure that if I don't know anything about botulism symptoms, but if they're similar, then yes, I think it's a very real possibility that botulism has been blamed for something that could well have been paralytic shellfish toxins. And I love it hearing you say that you eat the tunicates, and when people warn you about having tingling of your lips and so forth, because we've done some studies now, they're not yet published, with tunicates. They do take up saxitoxins. They do take them up to levels that are dangerous. And if you eat them, you probably are going to have tingling in your lips. So that is evidence that they could well be vectors, and that it's not something else like botulism, but is saxitoxin. You have anything to add?
I was just gonna echo what Don said. I'm not familiar with the symptoms of botulism, so I mean, it's a possibility that could have been PSP and not botulism, but
And to also, like Charlie, put people's mind at ease a little bit, I believe the Norton Sound Health Corporation has trainings annually now for their emergency crew and doctors, especially a state training regarding botulism and regarding paralytic shellfish poisoning, because we understand in this region how we can have both, and now, especially with this big bloom of 2022. So I believe that's my understanding, that the Norton Sound Health Corporation and our medical field has that going on as a result of all this. So that's also there to. Go ahead, Emma.
So Charlie, I think when the seafood processing plant was brought up that we have here, I think there was an idea of, can you give us all the bodies? So we can test them. There's a lot of data supporting that their toxins are not in the meat, which is why only the legs are sold.
Mhm.
I expect you will find someplace king crab, hepatopancreas that has toxins in it, because other crabs, it certainly happens.
So we have 2 questions in the chat. Claudia, I see your hand is up, but I'm assuming that's just because you haven't put
Yes.
it down.
Oh, yeah,
Okay.
sorry.
Okay.
I'm on.
That's all right. And so the first question is from Maggie Broadwater, and she writes, "Great talks, Don and Patrick. Can you share some information about potential subsistence food safety concerns?" So maybe we've already done this because she's been very patient. So that's the first one, is can you share some information about potential subsistence food safety concerns? Second is, what inferences can we make so far regarding appropriate risk communication with Bering Strait communities? Thank you.
well, with the
Well, first of all, hi Maggie,
Yeah.
and we wish you could have been here, and thank you for staying up so late.
Yeah, if you're on the East Coast, well,
Uh,
take tomorrow off.
thanks, Maggie. With regards to threats to subsistence resources, I think a question that comes up a lot is with at least the marine mammals like walruses and these northern fur seals is, are they safe to eat, or what are the risks with potentially eating these harvested animals? So with this northern fur seal event, while unfortunate, it actually provides us with some really interesting data. Now, this is just from 5 adults, so it's a small sample size, but these animals are confirmed saxitoxin poisoning event. So even at the time of death and being exposed to the amount of toxin that resulted in death, we did test a suite of the different tissues. So good news is that blubber and muscle and even brain don't seem to accumulate these toxins. So that is good to see. However, we are seeing moderate amounts of toxin in the kidney and liver. These are the tissues that are filtering out contaminants in your body. So it makes sense we do see saxitoxin specifically in the kidney, because that's what's filtering it out into your urine for excretion. So it's good to see that in terms of risk, it would be low risk to eat parts of marine mammals like these fat rich tissues like blubber and muscle and brain. Now with in terms of like eating clams out of the stomach of walruses, those are assumed to be clams taken from the environment, just like we take them from the environment with the Van Bean grab. So you see that those clams hold a lot of toxins. So those are things that would be potentially a higher risk to consume compared to eating, let's say, the blubber, mussel, and brain. And so there are some things that are some tissues that accumulate toxin. Again, this is a limited sample size, but then there are some other tissues that don't, which are good, like blubber and mussel and the brain. And so we are working on a tissue distribution study, looking at the suite of tissues in an animal that has been exposed to toxin and seeing where these toxins wind up for that purpose of consumption risk.
And the second part of her question was, what inferences can we make so far regarding, I guess that's appropriate risk communication with Bering Strait communities, was that?
Yeah, I think
You should answer that.
It was actually to both of you. So I
are part of the team.
I know, this is specifically to you. If Emma and I need to answer that for you, we can. I would say, inferences, I'm not sure what she's talking about on that one, but, so far regarding appropriate risk communication, I think in what we've learned, Emma Pate is here. What we've learned is that, the communication between all the research that's being done, as well as both at sea, as well as so at sea, when we had the bloom and there was an immediate communication, we wouldn't have been able to handle that or even know about it if we hadn't talked prior. And that, I have to say, was one of the first times I think we had crews, Dr. Picard, and had come ashore. I've met him before, and so I think, taking into account that he had to catch the boat in Nome and he was here and it's the university, I think that made a big difference as far as research communities being able to make it quickly get us information about risk communication, bringing us all along on this journey as you guys have found this problem to be in our neighborhood. And then, of course, I think that the Bering Strait region, I'll just say, has now having gone through the 2022 extreme bloom event. I think this Bering Strait region has done an amazing job. Everyone who is been or touched by this harmful algal bloom conundrum. So I think that we have, what would you say, Emma? You want to add anything to that? We are much further along as far as communicating risks when we get information from people. I would like to think that the people understand more about what is a harmful algal bloom and where to go to get information. Norton Sound Health Corporation, Nome, Eskimo, so forth, so on. Anything to add on that?
Awareness is definitely increased in 2022. When this bloom first happened, we didn't know what a HAB was or Alexandrium. We did a little bit because we actually started looking into doing baseline data collection for phytoplankton monitoring. However, we started that in February of 2020, just starting to look into it. March of 2020, we had the pandemic. 2021 was still a pandemic year. 2022 was the Alexandrium bloom, the massive bloom, the most toxic bloom, the longest persisting bloom in the US. So we had to figure things out and learn really fast.
Yeah.
So fortunately, we already started our trips to the communities around Nome to provide outreach information, education on what phytoplankton monitoring is, Alexandrium, Pseudonychia, Dinophysis, and So the communication foundation was somewhat there, but it was accelerated by the blooms in 2022 and 2023. And getting the real technical science out of the advisories to make it to where average individuals not knowing about HABs or phytoplankton monitoring can understand that. Our first advisory, we got complaints about there's too much science, it was hard to comprehend. So that was scaled back a little bit. And then adding in the services from Norton Sound Health Corporation to do the interpretations from English to Yupik, Siberian Yupik, etc. And that increased significantly. So because of the significant increase in information out there, we're able to downscale our advisories even more to be more direct because people are more aware and more informed. So as far as risks go, we can't say one way or the other if the food is safe to eat or not safe to eat. We're gathering data to help individuals make informed decisions for food security and food safety.
Yep, I think that's great. And actually Maggie just wrote, she's thinking in terms of blooms versus toxins in potential ocean foods. I think we're still learning.
I just wanted
That was awesome.
to add to what we just said and give some credit where credit is due. I have been part of this project, which has included Gay and Emma for a long time, so maybe I'm too close to it. Mike Brosnahan came in today and hadn't really seen a lot of this outreach or communication efforts. And he, after hearing a presentation from the two of you today, was honestly astounded or amazed by the complexity and the completeness of the information campaign that you two have done. And so the answer to part of Maggie's question is that, and when I compare it to us, we do all this in the Northeast, but there, the states close down the shellfish, people don't eat it, and they don't eat the crabs or whatever, and that's it. They don't worry about anything else. Here, you've got so much more to communicate, and you two, through the Norton Sound, when you work there from the Sea Grant perspective, have done an amazing job training people, having workshops, getting that information out, and it's out there now, and it's really impressive. So you deserve a lot of credit.
Well, thank you. Just leave it there. I think that reflects to the teamwork effort that goes on in the Bering Strait region. Yeah, that's how we deal with things. So thank you though for noticing. All right, we still have people on, and how are you guys holding up? We got questions, 2 more. And the last question for Maggie is, are there plans in the works for sampling by international partners to test the Gulf of Anadyr cyst-bed hypothesis?
Yes, the
Elusive.
The sad story there is that we actually obtained funding from the National Park Service for a collaborative project with Russian colleagues to do exactly that, for them to sample on the Russian side of the Chukchi Sea and the Russian side of the Bering Sea, to come to my lab, to bring the samples, to process them all in a uniform way. But that was shut down by the conflict in Ukraine. We can barely communicate with our colleagues there. I've had one Zoom call in, I think, 2 or 3 years now. So we wanted to do it. We had it all set up to do it, but we have not been able to make that happen.
So one suggestion to Maggie Broadwater, since you are, if you're still awake and you're in DC, is you might look into contacts you have with the State Department regarding trying to get at transboundary human health threats, shared marine ecosystem, commercial or otherwise, and subsistence threats to stocks, shared stocks in a transboundary region. I would just say that would be something I would start to look at. The State Department can handle that transboundary communicate. They are the appropriate federal entity to communicate federally to federally between the United States and the Russian Federation. So I think there's things there. There are other types of communications that go in under their purview regarding marine mammals and other things. So I think if you're in DC or want to talk about that more, that's where I would start to look, especially since you guys are federally attached, especially Maggie is with NOAA. So thank you very much for that question. Hope that helps. And then last but not least, Anjali Shah, you have been waiting patiently. I hope you're still on, Anjali, and she has a question about the walrus model.
Oh, yeah.
And that is, does the model take into account physical ocean mechanisms, especially the advective dynamics that cause the transported blooms?
Yeah, so good question. So this model, just to get into a little bit of the weeds, it's what we call a one compartment steady state model. So it's kind of an instantaneous exposure. So, and it assumes that there is no previous exposure. So, for example, the amount of cells in the water that we measured at each of those stations, along with the cysts at that point in time, or the amount of toxin we measure in those animals at that point in time, that is kind of what the model is based off of. That is one thing that we may try and simulate, which is simulating different bloom scenarios over different periods of time, and then looking at exposure over, if the bloom is 3 weeks, looking at how much toxin, if a walrus is foraging in an area for 3 weeks, how much accumulated toxin exposure are they exposed to? But that requires a bit more data and how quickly walruses eliminate the toxin, and some other things which we can estimate, but we just haven't got there yet.
Thanks for the answer. That makes a lot of sense.
All right, any other questions, comments for the dynamic duo it's been a while since we've had a 2-hour straight session, but they're still upright and rocking it. So if not, we will, before we close out though, I want to thank everybody for attending. Also, this is a double header week for Straight Science. So on this Friday, I'll start probably tonight putting out the notifications. We will be having a Straight Science on Friday. During 2023 and through 2025, there was a research project by Michigan State University and University of Alaska's Southeast looking at cruise ships in Nome. They were doing surveys with the ship tourists as well as residents of Nome. They are going to provide their results on Friday regarding cruise ship tourism in Nome. So that should be very interesting. And we will look forward to hearing their results. So with that, we'll thank you all, and we'll see you Friday. Have a good night, everyone, and be careful out there with the weather coming in.